Good afternoon, everyone. Welcome back to our class. Today's session is on chapter 12 from the book and chapter 6 in our class, which is on eukaryotes, or more specifically on, or with an emphasis on the fungi. All right. When it comes to pathogens coming from the eukaryotic domain, according to the WHO, almost over half of the world's population is infected with eukaryotic pathogens, and a part of these pathogens is the fungi play a very important role. All right, fungi are also beneficial apart from causing the infections. They are very important in a lot of industries, including the food chain, because one of their main characteristics is being a decomposer. So they decompose dead plants or dead plant matter, and thereby they recycle vital elements. The hard parts of the plants, which animals can digest, are decomposed primarily by fungi through the use of extracellular enzymes such as cellulase. Nearly all plants depend on symbiotic fungi, which is known as mycorrhizae, which helps their roots absorb minerals and water from the soil. Fungi farming ants cultivate fungi that break down cellulose and lining from plants, providing glucose that the ants can then digest. However, the humans use the fungi for food. Such a good example is the mushroom, and to produce other foods as well, such as bread, citric acid, and other drugs as well, such as alcohol or antibiotics, which is used against the bacteria. So the study of fungi is called mycology. A pathogen must be accurately identified to properly treat a disease and prevent its spread. We will first look at the structures that are the basis of fungal identification in clinical laboratory. Then we will explore their life cycle as well as the nutritional needs, and we will as well discuss on the benefits of fungi, which is used in many different, or economical benefits of fungi. So all fungi, in other words, are chemoheterotrophs, which means they require organic compounds for energy and carbon. So fungi are aerobic, they are facultatively anaerobic, and only few of the fungi are anaerobic, which we know. Okay, fungi colonies are described as vegetative structures because they compose of cells involved in catabolism and growth. So the thylus, which is the body of a mold or a freshly fungus, consists of long filaments, like you can see in the structure in the picture there. These long filaments are made of cells which they are joined together. So these filaments we call as hyphae, or in singular hypha. So hyphae can grow to an immense proportion. Using a DNA fingerprinting, some of the scientists, they mapped the hyphae of a single fungus in the United States, in the state of Oregon, and they extended it over 10.5 kilometers square, which if you put in or compare it with some other towns, it's around 10 times the Shahalam square, which, or in other words, is like if you know some very important squares in other countries. For instance, in Italy, we have the Piazza Santa Maria Novella square, which is a very big square and quite a tourist place. So what you see here in the picture 12.2a, in most molds, the hyphae contains cross walls, which we call them as septa. So you have a hyphae, and you can see that the cells, they are divided with these kind of like cross walls, and it's referred to as septa, or in singular we call it as septum, and which divides the cells into distinct and unnucleated or one nucleus cell-like units. So these hyphae are also called septate hyphae. When it comes to the part B in the picture itself, in few classes of fungi, the hyphae, it contains no septa and appear as long, continuous cells with many nuclei. So these are called choanocytic hyphae. And in the part C in there, even in fungi with septate hyphae, there are usually openings in septa that make the cytoplasm and adjacent cell continuous. These fungi are actually choanocytic organisms too. Hyphae grow by elongating at the tips. So each part of the hypha is capable of growth, and when a fragment breaks off, it can elongate to form a new hypha. Okay, in these pictures here, we have the aerial and vegetative hyphae. So in picture A, we have a photomicrograph of an aerial hyphae showing the reproductive spores. And the picture B, it's a colony of Aspergillus niger, which grows on a glucose agar plate, showing both vegetative as well as aerial hyphae. All right, yeast. Yeast actually are non-filamentous and unicellular fungi that are typically spherical, or sometimes they can be oval. Like molds, yeast are widely distributed in nature. They are frequently found as a white, powdery coating on fruits as well as leaves. Budding yeasts, such as Saccharomyces, um, they divide unevenly. So you can see in the picture in the figure 12.4, in a budding, the parent cells form a protuberance, which is a bud, on its outer surface. As the bud elongates, the parent cell's nucleus divides, and one nucleus migrates into the bud. Cell wall material is then laid down between the bud and the apparent cell, and the bud eventually breaks away. So one yeast cell can in time produce up to 24 daughter cells by budding. And some yeasts produce buds that fail to detach themselves. And these buds form a chain of cells, which we call them as pseudohyphae, because they resemble hyphae. And Candida albicans is one of the cells which attaches, one of the yeast which attaches itself to human epithelial cells as a yeast. But usually it requires pseudohyphae, or it requires the yeast to to invade those cells. So when it invades those cells, it upgrades itself into hyphae-like structure, and which is called pseudohyphae, and in this case is able to invade deeper tissues. When it comes to the fission yeasts, such as dividing, those are the yeasts which divide evenly to produce two new cells. During fission, the parent cells elongates, its nucleus divides, and two offspring cells are produced. So increases in number of yeast cells on a solid medium produce a colony similar to a bacterial colony. When it comes to dimorphic fungi, some fungi, most notably the pathogenic species, they exhibit dimorphism, which means two forms of growth. Such fungi can grow either as a mold or as a yeast. So the mold-like form produces vegetative forms. They produce vegetative and aerial hyphae, like yeast forms produced by budding. Dimorphism in pathogenic fungi is temperature dependent. At 37 degrees Celsius, the fungi is yeast-alike, whereas at 27 degrees Celsius, it's mold-like. What you can see in the picture there below. All right, we talk about it in a while. However, the appearance of the dimorphic in this instance is non-pathogenic. Fungus is shown in picture 12.5, which actually changes with CO2 concentration. When it comes to the life cycle, filamentous fungi can reproduce asexually by fragmentation of their hyphae. In addition, both sexual and asexual reproduction in fungi occurs by formation of spores. In fact, fungi are usually identified by spore types. So fungal spores, however, are quite different from the bacterial endospores. Bacterial endospores allow a bacterial cell to survive in adverse environment conditions, whereas when it comes to the fungi spores play a main role in asexual as well as asexual production or life cycle of the fungi. So asexual spores are formed by the hyphae of an organism. When the spores germinate, they become organisms that are genetically identical to the parents. Asexual spores are also produced by an individual fungus through the mitosis and subsequent cell division. There is no fusion of the nuclei of the cells. So there are two types of asexual spores which are produced by fungi, which is conidiospores and sporingiospores. All right, in this figure 12.6a, one type is a conidiospore or conidium, which is in plural is called as conidia. It's a unicellular or a multicellular spore that is not enclosed in a sac. Whereas the figure B, its conidia are produced in chains at the end of conidiospore. And such spores are produced by Penicillium or Aspergillus species. Conidia is formed by the fragmentation of the septate hyphae, which we mentioned earlier, into single and slightly thick-ended cells, which we call as atroconidia, which is another type of conidiospore called atroconidia. And in the picture C there, we have one species that produces such spores in Conidioides amitis, which is another species of conidiospore forming fungus. Another type of conidium is called blastoconidia, and they are formed from the buds of its parent cells. Such spores are found in some yeast, such as Candida albicans or Cryptococcus species. A Chlamydoconidium is a thick-walled spore, which is formed by rounding and enlargement within a hyphal segment. In the picture D there, or figure E, a fungus that produces Chlamydoconidia is the yeast, as mentioned earlier as well, is Candida albicans. So the other type of asexual spore, it's a sporangiospore, which is formed within a sporangium or a sac at the end of aerial hyphae, which we call it as a sporangiospore. Okay, on picture E there, the sporangium can contain hundreds of sporangiospores, and such spores are produced by Rhizopus. Okay, the sexual cycle or the sexual spores, they result from the fusion of nuclei from two opposite mating strains of the same species of fungus. Sexual spores require two different mating strains and so are made less frequently than asexual spores. Organisms that grow from sexual spores will have genetic characteristics of both parental strains. So we have three phases of sexual reproduction: plasmogeny, which is a haploid nucleus of a donor cell, penetrates the cytoplasm of a recipient cell. So the donor cell and the recipient cell nuclei, they fuse to form a diploid zygote nucleus. And then in meiosis, which has the diploid nucleus, gives rise to haploid nuclei or sexual spores, some of which may be genetic recombinants. So the sexual spores produced by fungi characterize the phyla. Clinical identification is based on microscopic examination of asexual spores, because most fungi exhibit only asexual spores in laboratory settings. Okay, nutritional adaptation. Fungi are generally adapted to environment that would be hostile to other bacteria. So fungi are chemoheterotroph, as we mentioned, and like bacteria, they absorb nutrients rather than ingest them as animals do. However, fungi differ from the bacteria in certain environmental requirements and in the following nutrition characteristics. So fungi, it's usually grow better in environments with a pH of 5, and which is like too acidic for the growth of most common bacteria. Almost all molds are aerobic, and most yeasts are facultative anaerobes. Most fungi are resistant to osmotic pressure than bacteria, and most can therefore grow in relatively high sugar or salt concentration. Fungi can grow on substances with a very low moisture content, generally too low to support the growth of bacteria. Fungi require somewhat less nitrogen than bacteria for an equivalent amount of growth. Fungi are often capable of metabolizing complex carbohydrate, such as lignine, which is a component of wood. Then most of bacteria, which can't use these compounds for nutrition. So these characteristics enable fungi to grow on such unlikely substances, such as bathroom walls, shoe leather, and discarded newspaper. Okay, this section provides an overview of medically important phyla or of fungi. So you note that only a relative small number of fungi can cause disease. So the genera named in the following phyla include many that are readily found as contaminants in food and in laboratory bacterial culture. Although these genera are not all of primary medical importance, they are typically examples of their respective groups. So they are divided into four, named as Zygomycota, Microsporidia, Ascomycota, and Basidiomycota. Referring to previous literature, there are many more groups. However, they have re-characterized the group and they have put into four most important. Zygomycota or conjunction fungi are saprophytic molds that have coenocytic hyphae, as we mentioned earlier. So they are produced sexually, which means forms when nuclei of the two similar cells. Right, so this is a diagram which is the life cycle of Rhizopus or zygomycete producing sexually as well as asexually. And asexual reproduction is through the sporangium or sporangiophore, as we mentioned, and sexual reproduction is using the two cells or the nucleus of the two cells. Okay, microsporidia are unusual eukaryotes because they lack mitochondria. Microsporidia also don't have microtubules, and they are obligate intracellular parasites. So it means they have to be inside the living cells in order to produce. And in early 1800s, when they were discovered, microsporidians were classified as fungi. They were reclassified later on as protists in 1980s because they lack mitochondria. And recent genome sequencing, however, reveals that microsporidians are no longer protists, but they belong to fungi. So nowadays they are considered as fungi. And here it's the life cycle of one of the microsporidians. We call it as Encephalitozoon intestinalis. It's an which causes a disease called as microsporidiosis, and it's an emerging opportunistic infection among the immunocompromised patients and the elderly. So the E. intestinalis, it causes diarrhea, and sexual reproduction has not been observed in this fungi. So it only produces asexually. Ascomycota, the other group, uh, Ascomycota, in other words, is called as well the sac fungi, which include molds with septate hyphae and some yeast. So their asexual spores are usually conidia produced in long chains from the conidiophore. So the term conidia means dust, and these spores freely detach the chain at the slightest disturbance and float in the air like a dust. And an ascospore forms when the nuclei of the two cells that can be either morphologically similar or dissimilar fuse. These spores are produced in sac-like structures called as ascus. So the member of this phylum are called sac fungi because of the ascus. So here we have life cycle of Thalaromyces, which is an ascomycete. Occasionally, when the two opposite mating cells from the two different strains, positive and negative, they fuse with each other, the sexual reproduction occurs. Okay, Basidiomycota, in other words, it's called club fungi, also possesses septate hyphae. So this phylum includes fungi that produces mushrooms. So basidiospores are formed externally on a base pedestral called as basidium. So the common name of the fungus is derived from the club shape of the basidium. There are usually 4 basidiospores per basidium. Some of the Basidiomycota, they produce asexual conidiospores. Fungal diseases Any fungal infection, it's called mycosis. So mycosis are generally chronic, which means long-lasting infections because fungi grow very slowly in comparison with other pathogens such as um, bacteria or virus. So mycosis are classified into 5 groups according to the degree of tissue involvement and mode, excuse me, to the degree of tissue involvement and mode of entry into the host cell. So we have the systemic, subcutaneous, cutaneous, superficial, or opportunistic. So fungi are related to animals. Consequently, drugs that affect fungal cells may also affect the animal cells, making fungal infection a human and other animals often difficult to treat. So fungi can cause disease by producing the toxins, similar to the bacteria as well. When it comes to systemic mycosis are fungal infections deep within the body. They are not restricted to any particular region of the body, but can affect a number of tissues and organs. So systemic mycosis are usually caused by fungi that live in soil. The spores are transmitted by inhalating or inhalation. These infections typically begin in the lungs and then they are spread around in other body tissues. Subcutaneous mycosis are fungi infections which are beneath the skin because which caused by saprophytic fungi that live in the soil and on vegetation. And sporotrichosis is a subcutaneous infection which is acquired by gardeners and farmers. It's quite often among them. And infection occurs by direct implantation of these spores that the fungi produces or mycelial fragments into a punctured wound in the skin. And fungi that infect only the epidermis or hair, nails are called as dermatophytes, and their infection are called dermatomycosis or cutaneous mycosis. And the fungi that cause superficial mycosis are localized along hair shafts and superficial epidermal cells. So these infections are prevalent in tropical climates. When it comes to opportunistic mycosis, an opportunistic pathogen is generally harmless in its normal habitat, but it can become pathogenic in a host who is seriously debilitated or traumatized, who is under treatment with broad spectrum antibiotics, whose immune system is suppressed by drugs or by an other immune disorder, or who has some type of lung disease. Okay, economical effects of fungi. Fungi have been used in biotechnology for many, many years. A type or species of fungi called as Aspergillus niger, for example, has been used to produce citric acid for food and beverage since early 1900s. And another type of fungi, a yeast called Saccharomyces cerevisiae, is used to make bread and wine. It is also genetically modified to produce a variety of proteins, including hepatitis B vaccine. And trichoderma is used commercially to produce the enzyme called cellulase, which is used to remove plant cell walls to produce a clear fruit juice. When the anti-cancer drug known as Taxol, which is produced by few trees, was discovered, there was a concern that the yew forests of the United States northern coast would be decimated to harvest the drug. So, however, the fungus Taxomyces also produces the drug Taxol. So fungi are used as biological control for pests as well. So in 1990s, the fungus Entomophaga unexpectedly proliferated and killed gypsy moths that were destroying trees in eastern United States. And the scientists, they are investigating the use of this type of fungi and many other several types of fungi which are able to kill the pests. So the fungus Conitherium militans feeds on fungi that destroy soya beans and other bean crops. A form which is filled with another species of fungi called P. formoceros is being used as well as biological alternative to chemicals in order to kill termites which hiding inside tree trunks and other hard to reach places. So in contrast to these beneficial effects, fungi can have undesirable effects as well for agriculture because of their nutritional adaptations. So I think with this slide, we can conclude today's chapter, and we will stop here to be continued with the next week on the lichens and water moths. So as for now, thank you very much and good luck.